Hello friends, I am Gaurav Makwana. I welcome you all to my another video tutorial of a Biotech guide. Today we are diving into one of the most fascinating foundation of a modern biotechnology, that is plant tissue culture media, its type and major constituents or a composition. So as usual, today's video going on a very interesting level. Stay tuned with us and enjoy this video lecture. So, what is culture media? Basically, for plant tissue culture, so is essentially an artificially prepared nutrient solution, or sometimes a gel that supports the growth and development of a plant cells, tissue, or even whole organs. What's the amazing is that all this happens under completely sterile and carefully controlled laboratory condition, giving us a total control over a plant mini environment. Now, what makes a perfect plant tissue culture medium? Well, it's all about getting the right balance. Here are the main characteristics every effective culture medium must have. First, balanced nutrient composition. All the essential macro and micro elements a plant needs. Second one is appropriate carbon source. Classic example is sucrose acting as a plant energy drink. Growth regulator or called hormone, special compounds that decide if the plant makes roots, shoots or jutes, bulk of the cells called callus. pH stability, the acidity or alkalinity of the medium must stay just right. Gelic agent quality, often agar for transforming the liquid medium into a supportive gel. Then supplementary organic additives, vitamin, amino acids that give extra boost. Sterility and aseptic handling, absolutely no contamination allowed. Species or stage specific formulation, because different plant and tissue might want different recipe. So these are the major characteristics which are required for a perfect plant tissue culture medium. Now let's break down some of the key media used in a plant tissue culture along with their history and main uses. First, we have White's medium, developed by Dr. White. Originally, it was used for root culture and is one of the earliest media types in tissue culture studies. It paved the way for future research in a plant cell growth. Second, MS medium. The Murashige and Skoog medium, designed to induce organogenesis and tissue regeneration. This is arguably the world's most widely used plant tissue culture medium. If you work in biotech, you will see MS medium in all kinds of a plant lab. Then there is a B5 medium created by Gemberg. This was made for cell suspension and callus culture, but its modified versions are now crucial for a protoplast culture, helping scientists isolate and study plant cells without walls. And 6 medium was introduced by Chu for most for cereal anther culture. Beyond cereals, it's found its way into all sorts of specialized tissue culture for a lab research. We also have niche medium by niche and niche. Originally for anther culture, it's still used today, especially for producing haploid plants from a pollen grains, a powerful tool for breeding. Stepping back from a specific discoveries, synthetic media include any media where all chemical components are strictly defined. These are now the standard for most tissue culture protocol, giving researchers precise control. Lastly, there is a natural media which contain a plant or a fruit extract and juices. Because their composition isn't well defined, they have become rare and are mostly replaced by synthetic media for a consistent result. So this is all about of our types of media. Now we are going to discuss our second main topic, composition of a media or a media constituents. So the composition of a plant tissue culture media can be divided into a few main categories. Let's break it down. Inorganic nutrients and organic nutrients. Inorganic nutrients, basically macronutrients and micronutrients are there. In macro, big players, nitrogen, phosphorus, potassium, calcium, magnesium and sulfur. And in micro, needed in a tiny amounts including iron, manganese, zinc, boron, copper and molybdenum. Then second, organic nutrients, carbon source like sugars, sucrose, glucose and other carbohydrates which fuels the cell growth. Then vitamins, essential helpers like thiamine, riboflavin and more keep cell functioning running. Then amino acid, building blocks such as a glutamine, asparagine and supporting various growth processes. Then undefined organic supplement, compounds like as a citrate, malate, succinate or fumarate sometimes give an extra edge. Then solidifying agents, substance like as a agarose, gel and gum turn liquid media into a gel, providing support to a plant tissue. Growth regulator, auxins, gibberellin, cytokine. Each group comes in a natural and synthetic form. Auxin promote root formation and cell division. Gibberellin mainly for a cell division and elongation. Cytokinin drives shoot initiation and a growth. So this classification provides a solid framework to understand what goes into plant tissue culture media. In next, we'll explore each category in detail. So don't forget to subscribe and stay tuned. Now let's uncover these all elements, inorganic and organic, one by one. First, macronutrients are the big building blocks plant need in a large amount, more than 0.5 grams per liter. Here's what they include: nitrogen, phosphorus, potassium, calcium, magnesium, sulfur. And don't forget to fundamental elements from air, carbon, hydrogen and oxygen. Quick tip for remembering the big six, just think never play king chess midnight shadow. N for nitrogen, P for phosphorus, K for potassium and like that. Now what do these nutrients do? They are essential for making up proteins, nucleic acid, cell membranes, vital enzymes and building up plant structure and metabolism. Basically, they are the bulk material that every plant cell draws from the thrive and multiply in your culture media. The International Association of a Plant Physiologists says anything above zero point 0.5 grams per liter is classified as a macronutrient. These are the major building blocks you can't ignore. Mastering them is the first step in creating an effective plant tissue culture medium. Second, micronutrients. These are needed in a very small traces, less than 0.5 gram per liter, but don't let their low quantity fool, because they are absolutely essential for a plant growth and metabolism. The main micronutrients you will find here: iron, manganese, zinc, boron, copper, cobalt and molybdenum. These micronutrients act as a catalyst and cofactor in vital enzyme processes and support major physiological function. Without them, a plant biochemistry would slow down or stop altogether. Always remember, micronutrients may be needed in a less than zero point 0.5 gram per liter, but they are crucial for a specific enzyme action and smoothly running biochemical reaction. And here you will remember by feeding man, zip, by careful controlled moves. Next, carbon source. Inside culture vessels, plants often lack chlorophyll or have a really weak photosynthesis. Add to that, there is a limited carbon dioxide and light, so the cells just can't make enough food by themselves. That's where external carbon sources come in. We need to add them to growth medium, and the most popular choice, sucrose, usually at a concentration of a 2 to 5%. When we autoclave the medium, that sucrose gets broken down into glucose and fructose, making it even easier for a plant cell to use. Sucrose does the double duty. It provides energy and keeps the medium osmotic pressure just right for a healthy cell growth. There are also other options like maltose, galactose, mannose, and lactose. Sometimes maltose is actually better for creating plant embryos in some culture. So this is all about of a carbon source. Without the right carbon source, plant tissue culture just wouldn't thrive. Next, vitamins. So in nature, plant synthesize their own vitamins, but in laboratory cultures, they usually can't make enough. This is why we need to add vitamins to our tissue culture media. Vitamins are essential for supporting enzyme function, which drives cell division and healthy development in a tissue culture. We can remember them using T and PM. Thiamine, absolutely essential for all plant cultures. Then nicotinic acid, vitamin B3, pyridoxine, vitamin B6, and myo-inositol, often called B vitamin and added in a larger amounts. Sometimes we also include pantothenic acid B5, vitamin C, ascorbic acid, vitamin D, and vitamin E. Vitamin C acts as an antioxidant, helping prevent the browning of tissue during handling. For stability, and thiamine, nicotinic acid, and pyridoxine are added in their hydrochloric form. Myo-inositol goes in at higher concentration, around 100 mg per liter. But there is the bottom line. Only thiamine and myo-inositol are strictly essential for a plant tissue culture media to work well. Next, a key player from the world of a plant tissue culture media, amino acids. Why do we sometimes add amino acids to the media, and what's their real role? First, amino acids provide reduced nitrogen in an organic form which plants can quickly and easily absorb. But in most cases, the inorganic nitrogen sources that we add to the medium, like ammonium NH4 and nitrate NO3, are actually enough to support plant growth. So we add amino acids only if there is not enough inorganic nitrogen available or if it's all been used up. A classic example is glycine, one of the simplest and most commonly used amino acids in this media. Another is cysteine, which works as an antioxidant, preventing tissues from turning brown by stopping unwanted oxidation. So this is all about of amino acids. Next, undefined organic supplement. Compounds like citrate, malate, succinate, and fumarate are not typical media ingredients. Their main role isn't in the medium. They are naturally involved in the metabolism happening inside plant cell, not as a direct media additives. Now let's talk about the three major gelling agents act as undefined organic supplement used to solidify culture media and support growing plant tissue. Agar, extracted from a red algae, typically used at concentration between a zero point 0.8 and 1%. It's widely popular, but the firmness can vary and acidic pH can partially break it down. Second, agarose. This is a purified form of agar used at about 0.4%. It forms a high strength gel perfect for delicate cultures that need extra support. Next, Galrit, made from a bacterial polysaccharides used at much lower concentration, around zero point 0.1 to 0.2%. It creates a pH stable, clear gel but requires careful stirring and sometimes leads to hyperhidrosity where tissues take up too much water. So this is all about of a undefined organic supplement. Next, last, quickly run through the main types of a growth regulator used in a plant tissue culture and why each one matters for a plant development in vitro. First, auxin. This promotes cell division and root growth. Common auxin include IAA indole 3 acetic acid, IBA indole 3 butyric acid, NAA 1 naphthalene acetic acid, 2,4-D 2,4-dichlorophenoxyacetic acid. So this auxin, IAA and IBA mainly help with a rooting and a shoot growth, especially when paired with a cytokinins, while 2,4-D is widely used to induce callus and somatic embryos. Next, cytokines encourage cell division and stimulate the formation of a shoots. The most popular examples are BAP6 benzyl aminopurine, kinetin, zeatin, and TDZ, thiazolone. Cytokines are a key for proliferating shoot and breaking bud dormancy, often working together with auxin for optimal growth. Gibberellin, preliminary gibberellic acid GA3 are not used as often, but they are crucial when you want to promote stem elongation and normal plantlet development. One thing to note, GA3 is heat sensitive, so it's added to the medium after sterilization. Ethylene is a special case. It's a gaseous hormone that mostly inhibit plant growth in a culture but can promote embryogenesis in some cases. Since plants naturally produce ethylene, it can build up inside culture vessel. Finally, abscisic acid. This regulator is essential for helping somatic embryos mature properly, closely mimicking how real seeds develop within a fruit. So each of these growth regulator has a specific role in guiding plant tissue development, rooting, shooting, elongation, or even embryo maturation. So this is all about of a growth regulator. Then how these hormones work together. Uh, High auxin to cytokine ratio leads to root formation. Then high cytokine to auxin ratio promotes shoot formation. And when both are in balance, that's an intermediate ratio, a mass of undifferentiated cells called callus forms. This simple rule is at the heart of a plant morphogenesis in the lab. Now, here, how you use this hormone in a practice. Auxin dissolve based in ethanol or dilute sodium hydroxide. Cytokines are dissolved in a dilute HCl, NaOH or DMSO if you are using TDZ. Gibberellin, like GA3, are filter sterilized and must be added after autoclaving your medium because heat destroys them. Ethylene management is also crucial. Too much can inhibit growth, so make sure there is good gas exchange or use adsorbents. And for those working with embryos, abscisic acid helps in the process of somatic embryo maturation. Each step ensures the right hormone content and a healthy plant culture. Let's wrap up of our exploration of types and constituents of the plant tissue culture media. If you found this video helpful, don't forget to hit the like button, share it with anyone who is interested in the plant biotechnology, and subscribe for more deep dives into plant science and lab techniques. Got question or want more content? Drop your thoughts in the comments. I'd love to hear from you. Thanks for watching, and I'll see you in the next video.